So today's topic is polysaccharides. The polysaccharides are made up of many repeating subunits of sugars or monosaccharides, subunits which are joined together by glycosidic linkages. These glycosidic bonds are of N-link type or O-link type. They are of two types, that is homopolysaccharides and heteropolysaccharides. Homopolysaccharide means that the repeating subunits of sugars are of same type, and if it is different sugar subunits are attached together or joined together by glycosidic bond, then it is a heteropolysaccharide. There are many types of, or the classes of, or the molecules which come under the category of polysaccharides. That is, they can be energy storage form or they can be structural components. In energy storage form, the first component is starch. Starch is a major energy storage form in plants. It is a homopolymer of glucose. It is synthesized in the chloroplast, and its precursor molecule is ADP glucose. Okay, this starch can be of two is the structure of starch is of two type, that is amylose and amylopectin. Amylose is straight chain and the bond is alpha 1,4 glycosidic bond. This amylopectin is branched and the bond is alpha 1,4 in straight chain and alpha 1,6 at branching points. And after how many subunits of sugar the branching comes, it comes after 25 to 30 residues of sugar. After every 25 to 30 residues there is branching. Okay, so the structure of starch is a helical structure. That means it is present in the form of helix, and the bond, if it is helix, then the bond is intra-chain hydrogen bonds in the structure of starch molecule. It is a non-reducing sugar. Why it is a non-reducing sugar? Because there is no free aldehyde or no free ketonic group, because all the free aldehyde and the ketonic groups are taking part in the bond formation. So there is no free aldehyde or ketonic group. That is why it is a non-reducing sugar. Okay, if there comes a question that there is n number of branching points, n number of branches, then how many reducing and non-reducing ends are present? So there will be only one reducing end and n plus 1 non-reducing end. Suppose if there is a question given that there are n number of branches, that is the number of branches is 2, that means the n is equal to 2. So how many reducing ends are there? There will be one reducing end and n plus 1, that means 2 plus 1 3 non-reducing end. Okay, so this was the structure of starch. The next one is glycogen. It is also a glycogen. What is glycogen? It is the major energy storage form in animals. In animals like us, it is the major energy storage form in animals. That is, we store all of our glucose in the form of glycogen, and where we store it? We store it in our liver and muscles. Okay, in our body there is almost 200 to 300 gram of glycogen present. Okay, it is also synthesized by bacteria and fungi. It is also a non-reducing sugar because it has no aldehyde or ketonic groups which are free. its structure is helical structure. If it is a helical structure, that means that it has intra-chain hydrogen bonds. Okay, same type of calculation over here also, that if there are n number of branches, then there is one reducing end and n plus 1 non-reducing end. It is also a homopolymer of glucose with same linkages. That means alpha 1,4 linkage and in the straight chain and alpha 1,6 linkage at branches, and it is more branched than starch. That means कि after this branching appears after every 10 sugar residues. Okay, this was about the energy storage form in animals, that is glycogen. The next energy storage form is inulin. Okay, inulin, it is a homopolymer of fructose and the linkage is beta 2,1 linkage. That means the glycosidic bond is beta 2,1 linkage. Its structure is also helical. If it is helical, that means intra-chain hydrogen bonds. Okay, it is also unbranched and it is also a non-reducing sugar. Non-reducing sugar means no free aldehyde or ketone group is present. This inulin is not digested by majority of animals blood. Okay, that means if it is given, if inulin is given in the form of injection, then most of the inulin is excreted out. Then this excreted portion of inulin can be used to detect the GFR. GFR is glomerular filtration rate. It is used in the analysis of GFR, and this inulin is considered in GFR as a gold standard. Okay, so this was about the polysaccharides which are used as an energy storage form. In the next video, we will discuss some other polysaccharides which are playing important role in structural formation like cellulose, chitin, pectin, and about heteropolysaccharides also. If you like this video, subscribe my YouTube channel and hit the like button. Thank you for watching.